oxidized in a pathogenesis context. So we grew Vibrio cholerae under a virulence-inducing condition, then treated with an organic ROS. Then we collected the cells. Here I'm showing you an example protein containing cysteine residues with thiols in possible oxidation states. It may remain reduced as a sulfhydryl group, oxidized to form disulfide bonds, or have other oxidation modifications. So we first treat with N-ethylmelamide. So we first treat with N-ethylmelamide to block any free thiols in the reduced state. And then we treat the sample by DTT, reducing the originally oxidized cysteine residues. Pass the sample through a thioenriching column and digest the sample into small peptides by trypsin, and elute the free thiol-containing peptides from the column. And then we label these once oxidized thiols by idoacetamide, which adds a 57 Dalton carbomidyl methyl group to the thiol group. And we then submit the samples to mass spec analysis and look for peptides containing cysteine with a carbomidyl methyl modification. So we identified 266 Vibrio cholerae proteins containing oxidized cysteine residues, including some of the ROS-resistant proteins I showed you earlier, such as peroxyredoxin A and catalase G. But we're most interested in proteins in this group, regulator proteins. One of the hits in this group is APHB, a virulence activator that turns on virulence when sensing a microaerobic environment. Previous work in the lab demonstrated that APHB achieves this redox sensing through the redox reactive cysteine residue 235. In an oxygen-rich environment, C235 is oxidized, and APHB exists as monomers. Under low oxygen conditions, C235 becomes reduced, APHB dimerizes and activates virulence, mutating the cysteine to serine. And mutating the cysteine to a serine, an amino acid that are sterically similar to cysteine but cannot be oxidized, made this mutant string more susceptible to both inorganic and organic hydroperoxide, as shown here by the enlarged inhibition zones. These cleared rings without bacterial growth around a central dot releasing ROS. So the importance of the APHB C235 for both virulence as well as ROS resistance motivated us to study more regulator proteins containing redox sensitive cysteine. So these two peptides containing a carbomidyl methyl modification and cysteine residue is signified by a 160 Dalton difference between species B5 and B6, B5 and B6 and Y5 and Y6 here on the spectrum, for example, also caught our attention. These peptides are from another regulator protein, ArcA. Looking at the amino acid sequence of ArcA, we can see that these two cysteine residues in the 173rd and 233rd positions are its only cysteine residues. So what is ArcA, you may wonder? ArcA is the response regulator in the anoxic redox control, or some other people call it aerobic respiratory control two-component system. This two-component system serves as a global switch that adapts bacteria to different redox environments. When sensing a more reduced quinone pool at the inner membrane, the first component, the sensor histidine kinase, ArcB, autophosphorylates. This phosphorylate group is relayed onto an aspartate residue on ArcA, activating the response regulator. The activated ArcA then goes on to bind to the DNA, leading up to the genes regulated by ArcA. Repressing genes that are involved in aerobic respiration, such as those in the TCA cycle, and activating genes that are involved in a more anaerobic lifestyle. The ArcA protein mainly consists of two parts: the receiver domain, which receives signal from the histone kinase contains the phosphorylation site aspartate 54, abbreviated here, and the rest of the talk as D54. And the DNA binding domain, which contains both of the cysteine residues that we identified in the proteomic work, C173 and C233. So to confirm the mass spec result, I used this similar thiol labeling approach to helicell lysates expressing his-tagged RK proteins. The normal version of RK, or as we call it, the wild-type form, and cysteine to serine mutant versions with one or both cysteine residues mutated to a serine. Any oxidized thiol would be labeled by a 0.5 kilodalton AMS, and with this added weight, would migrate slower on an SDS page gel and show up at a higher position compared to an unlabeled version. As you can see, in the absence of ROS, all versions of RK showed up at the original RK size, 28 kilodaltons, suggesting that they do not have any oxidized thiols under this growth condition. In the presence of ROS, the double cysteine mutant without any cysteine migrated to the same position as the untreated samples, suggesting an absence of oxidized cysteine residues, apparently. In contrast, both C173S and C233S migrated to a slightly higher position. Since both of these single cysteine mutants each only contains one cysteine, their size shift suggests the other cysteine residue was oxidized in the ROS treatment. The wild-type sample showed up at an even higher place, suggesting multiple modifications, multiple cysteine oxidation. Taken together, we've confirmed the result from the proteomic screening that both cysteine residues of RK are oxidized when bacteria is challenged by ROS. So I constructed the RK null and ArcB null mutants by disrupting them with antibiotic resistance genes. And I put back on the Vibrio cholerae chromosome on the RK gene with its native promoter on the LAC-Z locus. LAC-Z encodes a beta-galactosidase that does not affect Vibrio cholerae physiology. To separate the redox reactive functions of the cysteine residues from the rest of RK functions, as you may recall, RK is a global regulator that does a lot of different things. I also put back in the LAC-Z locus RK variants with each one or both cysteine residues changed to a serine. I also made a unphosphorylatable version where I changed the aspartate residue at the phosphorylation site to an alanine. And to keep the genetic background consistent, these strains also are also in LAC-Z minus background. So we quickly see that RK mutants grow poorly, as shown in the red line here. C173S mutants grew similarly to wild type, this green line here, and C233S mutants grew more similarly to RK mutants. And by spotting serially diluted bacterial samples on an LB plate at a low enough dilution, like here on the 5th tenfold dilution, you'll get these single visible dots after an overnight growth. Each of these dots are a single colony and are derived from a single bacteria. If you look at the colony size of these strains, we can quickly notice that RK mutants is much smaller compared to wild type. The complementation and C173S mutants are normal sized, but all the other mutants have small colonies, just like RK mutants. One of the first described phenotypes of an RK mutant is its sensitivity to certain dyes, such as toluidine blue. Indeed, when we spot RK mutants on an LB plate containing toluidine blue at a concentration that doesn't affect wild-type growth, RK is at least 3 logs more sensitive. Again, while RK wild-type and C173S at the LAC-Z locus restore TB survival, these other mutants showed sensitivity to the dye similar to RK mutants. When grown under microaerobic conditions that is known to activate RK functions, again, the RK null mutant and C233S mutants have less growth, while the complementation string and C173S mutants grew normally. Since C233S showed a similar growth effect to that of an ArcAB null mutant, the cysteine to serine mutation at this position may have affected the structure of ArcAB and its fundamental functions. But luckily, we still have C173 that may help us tease apart the redox cysteine related functions of ArcAB from the rest of ArcAB functions. Since cysteine residues are oxidized in oxidative stress, I want to see how a cysteine to serine mutant behaves upon ROS exposure. Although C173S mutant did not affect normal growth in ArcAB activating conditions, when challenged with ROS, C173S mutants are about 30-fold more sensitive compared to wild type, suggesting an important role for C173 in ArcAB functions during ROS stress. So, as a response regulator, ArcAB's main function when activated is to bind to DNA sequences that have the code to summon ArcAB binding. This DNA code or ArcAB binding box is shown here. SDHC, which encodes succinate dehydrogenase, has a predicted ArcAB binding box in its promoter region. It is known to be repressed by phosphorylated ArcAB in E. coli. We put this specific ArcAB box controlling SDHC expression in front of a LAX-E gene encoding a beta-galactosidase on a plasmid, then we can quantify the expression of SDHC as beta-galactosidase activities from different strains hosting the reporter plasmid. Under a microaerobic growth condition and ArcAB activating condition, there is little SDHC expression in the wild type, suggesting active ArcAB repression. In an ArcAB null mutant, the lack of ArcAB protein, therefore a lack of ArcAB repression, resulted in high levels of expression. The C173S mutant showed almost the same level of repression as wild type. But when challenged by ROS, we see a slightly elevated SDHC expression in wild type, but repression is mostly retained. However, the C173S mutant showed a much weaker repression, as signified by the higher expression level here. So next, I wanted to test if this in addition, I also tested if ArcAB cysteine mutation affect ROS resistant gene expression. OHRA encodes an organic hydroperoxide resistance protein. Previous work in the lab has shown that OHRA has a cysteine containing regulator OHR that de-represses upon oxidation. We checked how ArcAB affect OHRA expression by qPCR. We see ROS inducing OHRA expression in wild type, consistent with previous work in the lab. However, in the C173S mutant, OHRA expression remains low even when ROS is present, suggesting a possible lack of resistance protein in C173S mutants to provide ROS protection. So this OHRA is also important for Vibrio cholerae resistance to ROS during colonization of the mammalian host. Our lab has previously developed an infection protocol for adult CD1 mice with the ROS scavenger N-acetylcysteine. We showed that without NAC treatment, the mouse gut is a ROS-rich environment, as signified by the fluorescent signal from the ROS dichlorofluorosine diacetate. With NAC added in the drinking water, the ROS in the mouse gut is significantly removed. Co-colonization with wild-type Vibrio cholerae and OHRA mutant lacking the ROS-resistant protein in the untreated mice showed a significant colonization defect in OHRA mutants. As you can see here, the competitive index, which is calculated by the mutant colony forming unit over the wild type, is way below 1. When co-colonizing mice treated with NAC, or in the ROS mitigated guts, OHRA mutants colonized almost as well as wild type, as indicated here by a competitive index close to 1, suggesting an important role for OHRA in ROS resistance during infection. This method gives an opportunity to tease apart the ROS-related aspects from other factors during colonization. So I used a similar protocol where I took 2 groups of mice, one without NAC treatment with normal levels of gut ROS, and one with NAC added to their drinking water, removing much of the ROS, and co-colonize each group with a 1-to-1 mix of the C173S mutant and the wild type Vibrio cholerae. At these respective time points, I enumerated wild type and mutant Vibrio cholerae recovered from mouse poop by plating on LB plates containing X-gal. Wild type with a functional LAC-Z gene would make blue colonies on these plates, and my mutant would be white. So we can see that in the untreated mice, or in the ROS-rich guts, the C173 mutants showed more severe defects as time progresses, whereas in the ROS-mitigated guts, the C173S mutant colonizes almost as well as wild type, even after 6 days. This data suggests that C173 residue on RK is important for colonization in an ROS context. So here is a summary of what I've told you so far. I've shown you that both of the cysteine residues on RK can be oxidized upon exposure of the cells to ROS. The RK null mutant, C233S mutant, the double cysteine mutant, the phosphorylation mutant, and the ArcB null mutant all have growth defects, suggesting important function of this 2-component system in general, and that C233S mutation might have compromised the structure of RK. The C173S mutant grows just as well as the wild type under normal conditions, but upon exposure to ROS is much more sensitive. This mutant cannot carry out normal regulatory functions of RK under ROS stress, and they fail to properly respond to ROS stress by failing to amp up the production of resistance proteins. And they can colonize just as well as the wild type in ROS removed guts, but has a colonization defect in ROS rich guts. So why is RK, so why is the ability to be oxidized as a cysteine residue so important for RK functions and ROS response? So like I mentioned earlier, the sulfhydryl group on the cysteine residue is highly reactive to redox changes. And the redox state of the Vibrio cholerae APHB changes the regulatory behaviors of APHB. And we have seen in similar examples in other bacteria where oxidation at cysteine residues alter the regulatory behaviors of transcription regulators. For example, the regulator AGRA can no longer bind to DNA upon formation of the intramolecular disulfide bond. The Burkholderia thailandensis regulator BIF-R binds more tightly to DNA upon formation of the intermolecular disulfide bond. And RIT-R, also a response regulator, undergoes a conformational change that opens up its receiver domain from the DNA binding domain, activating its DNA binding activities. So therefore, I hypothesized that RK cysteine residues are redox sensing, and oxidation affects RK regulatory functions such that RK function is retained during ROS stress. One possibility is that oxidation makes phosphorylation easier, activating RK activities. During ROS insults, oxidation at RK cysteine residues might change the, my internet connection is unstable, sorry people. Oxidation at RK cysteine residues might change the shape of RK, making the phosphorylation more accessible. To test this, I visualized RK phosphorylation by carbometyl phosphate on phosphatic gels. Phosphatic gels are just SDS page gels containing phosphatags and divalent cations, manganese in my case, that together have high affinity to phosphorylated proteins. Phosphorylated proteins migrate slower on a phosphatic gel due to trapping by these molecules. So I added an increasing concentration of carbometyl phosphate to either previously reduced or oxidized RK. If oxidation of RK facilitates easier phosphorylation, I would see phosphorylated RK at a lower carbometyl phosphate concentration with oxidized RK compared to the reduced RK. However, this is not the case. As you can see on this phosphatic gel, both forms of RK require the same amount of carbometyl phosphate to be phosphorylated. So when I tried to look at RK phosphorylation states with host cell lysates of Vibrio cholerae grown under RK activating microaerobic conditions, I see RK phosphorylated in both wild type and ArcAB mutants. This suggests that RK can receive positive regulation from more than just its cognate kinase ArcAB. And when phosphorylation site is missing D54A mutant, no phosphorylation of RK is seen. There's no band here, suggesting D54 is absolutely required for RK phosphorylation. And when wild type is challenged by ROS, the phosphorylated species is also gone, suggesting an external ROS exposure abolishing RK phosphorylation. So this is curious because we did see wild type repressing SDHC expression during ROS stress, suggesting functional RK. If oxidation is not helping phosphorylation, maybe it's stepping in to activate RK functions instead of phosphorylation. In other words, we hypothesize oxidation mimics phosphorylation. To test if oxidation can activate RK binding similar to phosphorylation, I performed electrophoretic mobility shift assays with purified RK variants under different treatments. Sure enough, with the wild type RK, we see a DNA protein complex forming when the protein is being phosphorylated or oxidized, as signified by the higher position here due to slower migration, but not in its unphosphorylated reduced form when it's not activated. With the C173S mutant protein, although we see DNA binding when it's phosphorylated, it can no longer respond to oxidation. To further test if this oxidation activation of DNA binding activity depends on phosphorylation, I tested the same conditions on a phosphorylation mutant protein D54A. Although it does not bind to DNA under phosphorylated conditions, since it can no longer be phosphorylated, it showed binding when oxidized. These data suggest that oxidation can activate RK's DNA binding activity, just like phosphorylation. And this does not depend on phosphorylation. And C173 is required for this oxidation-mediated activation. So what are these post-translational modifications doing to RK to activate its DNA binding activities? One hypothesis is that either of these processes, phosphorylation or oxidation, increases RK-RK protein interaction, which leads to activation. One commonly used methods to detect protein-protein interaction is a bacterial 2 hybrid system. This system takes advantage of 2 catalytic domains of an adenylate cyclase, the T25 and T18 domains, that need to be in close proximity to function. In an adenylate cyclase mutant, the interacting partners in question are translationally fused to each one of the catalytic domains. When the proteins in question interact, they bring the 2 domains together, generating cyclic AMP that activate the transcription of a reporter gene. By quantifying the expression levels of the reporter gene, in this case, again, beta-galactosidase activities, we can assess the level of protein interaction. So I first tested if a microaerobic growth condition promotes ArcAB interaction. Indeed, I saw a higher beta-galactosidase activity compared to when I grew cells aerobically with one of the interacting partners changed to a C173S mutant, microaerobic growth still promotes ArcAB interaction, consistent with our previous gel shift results. So what about oxidation? We can see that the ROS treatment caused an increase in ArcAB interactions compared to the untreated cells with the wild-type form of ArcAB as interacting partners. But when C173S is involved, I can It
looks like the screen sharing.
I don't know what happened. Okay, is my audio still on? Okay, yes, it is. Okay, let me put this. Okay, sorry about that. Okay, so what about oxidation? We can see that ROS treatment caused an increase in our RK-RK interaction compared to the untreated cells with wild-type form of RK as interacting partners. But when C173S is involved, I can no longer see these ROS-induced interaction. So these results suggest that oxidation does promote RK-RK interaction, and the oxidation-dependent interaction is dependent on C173. So what does increased RK-RK interaction suggest? The Stock lab at Rutgers has shown with an analytical size exclusion chromatography that E. coli RK forms dimers and tetramers when phosphorylated, but they found it was impossible to do further solution studies as phosphorylated RK falls out of solution. I ran into the same problem with Vibrio-cholerae RK. Here I'm showing you the percentage of proteins that remained in the supernatant after centrifugation. When phosphorylated, RK can't even sustain a standard tabletop centrifuge spin, whereas the reduced unphosphorylated and oxidized unphosphorylated forms are mostly soluble even at very high centrifugal forces. With the soluble fractions of each of these in vitro treatment, I tried to determine oligomeric state changes of Vibrio-cholerae RK by size exclusion chromatography. The tiny portion of phosphorylated RK shown in bright blue here eluted at the same volume as the unphosphorylated control shown in purple here. However, oxidized RK showed two peaks, first eluting at much earlier volume corresponding to a much larger complex, then again at the RK monomer volume. This shift in retention time suggests that oxidation can change RK oligomeric states. So we further investigated the effect of oxidation on RK by running a non-reducing SDS page gel with cell lysates growing microaerobically in a combination of ROS and the reducing agent beta-mercaptoethanol, or BME. We see that RK forms an intramolecular disulfide bond upon ROS exposure, as signified by the slightly lower band here compared to its original size. This species is gone when reducing agent is included in the growth media, suggesting that this is indeed oxidation mediated. In single cysteine mutants, the lack of the other cysteine to form the intramolecular disulfide bond drives these RK variants to form dimers, especially in the C173S mutants. A lot of dimers are formed with a C233 mediated intermolecular disulfide bond. We can see that with the double cysteine mutant that these oxidation mediated changes like this one and these dimers are not present. So all of these are indeed cysteine mediated. So the data I've shown you so far suggests the following working model. In an oxygen-rich environment, RK, a cytosolic protein, is in the reducing cytoplasm, unphosphorylated, and in its inactive state as monomers. Under microaerobic conditions, RK is phosphorylated, facilitating RK-RK interactions, enabling DNA binding and RK regulatory functions. Under ROS stress, RK phosphorylation is abolished, but C173 and C233 are forming a disulfide bond, introducing a conformation that promotes RK-RK interactions, DNA binding, and retaining RK activities, at least partially. If we take a look at other response regulators with similar DNA binding structures, these cysteine residues are unique to RK. This is in alignment of the DNA binding domain sequences for several response regulators, with the C173 position showing red and the 233 position in blue. Other response regulators in the same family do not have these cysteine residues. My speculation is that having redox sensing cysteine residues in RK, the response regulator that adapts the bacteria to a different oxygen level, it serves two functions at the same time: adapting the pathogen to the oxygen limiting condition and also the ROS-rich environment, that is the host. In fact, RK cysteine residues are conserved in many other bacteria, including other enteric pathogens such as E. coli and Salmonella. So as a proof of concept, I tested if the mechanism we see in Vibrio cholerae is also important in other enteric bacteria. I constructed mutant and complementation strains in Salmonella enterica serovar typhimurium. Similar to what's seen in Vibrio cholerae, Salmonella C173S mutants are also more sensitive to in vitro ROS challenge. And when infecting mammalian cells, although initial uptake was similar, C173S mutants showed much less intracellular replication compared to wild type. So to summarize the second part, RK phosphorylation is abolished in Vibrio cholerae upon extracellular ROS exposure. In vitro, oxidized RK protein can bind to target DNA similar to that of phosphorylated RK, and thus oxidation sensing depends on C173. I have shown you with a bacterial to hybrid system that both phosphorylation and oxidation increase RK-RK interactions. And again, the oxidation-dependent RK interactions requires C173. And oxidation can change RK's oligomeric state and induces the formation of an intramolecular disulfide bond. Taken together, we propose that oxidation at the cysteine residues results in a functional mimic of a phosphorylation activated RK, which retains RK activity under ROS stress. And this mechanism is probably widely used by enteric pathogens to adapt to the host environment. I hope by this point, by showing you this bacterial oxidative stress response mechanism, you're impressed by the versatile and practical designs from prokaryotes, who are just trying their best to adapt and survive in even the worst case scenario. And with that, I want to acknowledge all the entities that made this work possible. I'd like to thank my thesis committee for their scientific insights and tremendous mental and material help. I want to thank Stefan and Michael for their generous help with sizing columns. Members of the Shin Lab and the Brodsky Lab have taught me how to make salmonella strains and do infection assays with cell cultures. Ann Stock and Rongao from Rutgers have kindly provided key insights about RK structure. My high school friend Hao Yangzhen pointed me to the resources to scan the Vibrio cholerae genome for an RK motif. I'd like to thank my neighbors and staff in Johnson Pavilion for a supportive work environment. I would also like to acknowledge my home department in biology, Colleen, Robin, Mike, and Fevzi had made sure that I had the help I needed when I needed it. And Q, I don't think I've ever met in person, have bought me all the gadgets, monitors, external keyboards, all the things that made my life easier. I am appreciative of the incredible members of the Jewel Lab and the Goulian Lab for our joint lab meeting discussions, their generous trainings, and our friendships. I am greatly thankful for my advisor, Jay, for all the patience and guidance he offered me for my project and my development as a researcher. He embodies a passionate and resilient scientist with the strongest work ethic imaginable. Being the only grad student in the lab is kind of like being an academic only child, but Jean Schulte is like the best academic cousin anyone could ask for. She has lent me her keen scientific perspectives, calm demeanor, and company throughout these years. I also enjoyed company from the Shin Lab next door. They're always exuberating happy vibes. I am lucky to have experienced the tail end of BioBall, which was just biology grad students playing basketball at 8 a.m. for an hour. And also the tail end of fall semester finals week wreath making at the greenhouse. That was very therapeutic. I want to commend my cohort for their accomplishment and our quality porch time, board game, and movie nights. Next, I want to thank all of my friends who are major parts of my support network. Thanks for putting up with me being ridiculously late because of hiccups in the lab. Thanks for hanging out with me in the lab, taking me onto culinary adventures, and letting me pet your dogs. Lastly, I want to thank my family. I am lucky to be in a PhD program close to my parents. Knowing they are there for me just an hour away is invaluable. I truly appreciate their unconditional trust in me. My family physically further away have also offered help in various ways. I really appreciate all of that. I want to credit my boyfriend, Greg, who's actually in this room with me right now, my only audience here in 209. I want to credit him for the technical, logistics, and emotional support throughout the duration of this work. I am greatly motivated by love of Greg and his family. And with that, I want to take any question you might have. Just unmute yourself, or I read the question in the comments, probably. Let me find the chat. Maybe I stop sharing the screen. Yeah. Oh, true.
Nobody said.
Oh, nobody said anything yet. Did I present? Did I? Okay.
might put some people on.
Okay.
Your computer is acting.
I think so, yes.
Oh,
Thank you, people.
yeah, then let's go to the other room to continue our defense.
Okay, then now my committee will grill me privately, so we'll log off for now. I'll
Thank
send
you,
out an
everyone.
email if there's
Great
a
job.
solid. Thank you. Hey, Kylan.
See you
All right,
soon.
I'll log off from this one now. Thank you, people.